Ricketsia is a genus of nonmotile, gram-negative, non-spore-forming, highly pleomorphic bacteria that can be present as cocci, 0.1 micrometer in diameter, rods, 1 to 4 micrometers long, or thread-like, 10 micrometers long. The term Ricketsia, named after Howard Taylor Ricketts, is often used interchangeably for any member of the Rickettsiales, being obligate intracellular parasites. The Ricketsia survival depends on entry, growth, and replication within the cytoplasm of eukaryotic host cells, typically endothelial cells. Ricketsia cannot live in artificial nutrient environments and is grown either in tissue or embryo cultures. Typically, chicken embryos are used, a method developed by Ernest William Goodpasture and his colleagues at Vanderbilt University in the early 1930s. Ricketsia species are transmitted by numerous types of arthropod, including chigger, ticks, fleas, and lice, and are associated with both human and plant disease. Most notably, Ricketsia species are the pathogen responsible for typhus, Rickettsialpox, Boutonneuse fever, African tick bite fever, Rocky Mountain spotted fever, Flinders Island spotted fever and Queensland tick typhus, Australian tick typhus. Despite the similar name, Ricketsia bacteria do not cause rickets, which is a result of vitamin D deficiency. The majority of Ricketsia bacteria are susceptible to antibiotics of the tetracycline group. Classification. The classification of Ricketsia into three groups, spotted fever, typhus, and scrub typhus, was initially based on serology. This grouping has since been confirmed by DNA sequencing. All three of these contain human pathogens. The scrub typhus group has been reclassified as a related new genus, Rickettsia, but many medical textbooks still list this group under the rickettsial diseases. Ricketsia are more widespread than previously believed and are known to be associated with arthropods, leeches, and protists. Divisions have also been identified in the spotted fever group, and this group likely should be divided into two clades. Arthropod inhabiting Ricketsiae are generally associated with reproductive manipulation, such as parthenogenesis, to persist in host lineage. In March 2010, Swedish researchers reported a case of bacterial meningitis in a woman caused by Ricketsia helvetica, previously thought to be harmless. Spotted fever group, Ricketsia Ricketsiae, Western Hemisphere, Rocky Mountain spotted fever, Ricketsia ricari, USA, former Soviet Union, Rickettsialpox, Ricketsia konoi, Mediterranean countries, Africa, Southwest Asia, India, Boutonneuse fever, Ricketsia sibirica, Siberia, Mongolia, northern China, Siberian tick typhus and North Asian tick typhus, Ricketsia australis, Australia, Australian tick typhus, Ricketsia felis, North and South America, Southern Europe, Australia, flea-borne spotted fever, Ricketsia japonica, Japan, Oriental spotted fever, Ricketsia africate, South Africa, African tick bite fever, Ricketsia hoogstrali, Croatia, Spain and Georgia USA. Unknown pathogenicity. Typhus group, Ricketsia prowazeki, worldwide, epidemic typhus, recrudescent typhus, and sporadic typhus, Ricketsia typhi, worldwide, murine typhus, endemic typhus, scrub typhus group. The causative agent of scrub typhus, formerly known as R-Suit sugarmushi, has been reclassified into the genus Rickettsia. Flora and fauna pathogenesis. Plant diseases have been associated with these Ricketsia-like organisms. RLOs. Beet latent rosette RLO. Citrus greening bacteria and possibly this citrus greening disease. Cloverleaf RLO. grapevine infectious necrosis RLO, grapevine pierces RLO, grapevine yellows RLO, witch's broom disease on larynx SPP, peach phony RLO, papaya bunchy top disease. Infection occurs in non-human mammals. For example, species of Rickettsiales have been found to afflict the South American guanaco, llama guanaco. Pathophysiology. Genomics. Certain segments of Ricketsial genomes resemble those of mitochondria. The deciphered genome of R. Prowazeki is 1,111,523 BP long and contains 834 genes. Unlike free-living bacteria, it contains no genes for anaerobic glycolysis, genes involved in the biosynthesis and regulation of amino acids and nucleosides. In this regard, it is similar to mitochondrial genomes. In both cases, nuclear host resources are used. ATP production in Rickettsia is the same as that in mitochondria. In fact, of all the microbes known, the Rickettsiale is probably the closest relative, in a phylogenetic sense, to the mitochondria. Unlike the latter, the genome of R. Prowazeki, however, contains a complete set of genes encoding for the tricarboxylic acid cycle and the respiratory chain complex. Still, the genomes of the Rickettsia, as well as the mitochondria, are frequently said to be small, highly derived products of several types of reductive evolution. The recent discovery of another parallel between Rickettsiales and viruses may become a basis for fighting HIV infection. Human immune response to the scrub typhus pathogen, Orientia sugamushi, appears to provide a beneficial effect against HIV infection progress, negatively influencing the virus replication process. A probable reason for this actively studied phenomenon is a certain degree of homology between the Ricketsiae and the virus, namely, common epitope, S, due to common genome fragment, S, in both pathogens. Surprisingly, the other infection reported to be likely to provide the same effect, decrease in viral load. Is the virus-caused illness dengue fever? Comparative analysis of genomic sequences have also identified 5 conserved signature indels in important proteins which are uniquely found in members of the genus Rickettsiales. These indels consist of a 4-amino acid insertion in transcription repair coupling factor MFD, a 10-amino acid insertion in ribosomal protein L19, a 1-amino acid insertion in FDSZ, a 1-amino acid insertion in major sigma factor 70, and a 1-amino acid deletion in exonuclease 7. These indels are all characteristic of the genus and serve as molecular markers for Rickettsia. Naming. The genus Rickettsia is named after Howard Taylor Ricketts, 1871–1910, who studied Rocky Mountain spotted fever in the Bitterroot Valley of Montana, and eventually died of typhus after studying that disease in Mexico City. Rickettsiale genomes and related information at Patrick, a bioinformatics resource center funded by NIAID. African tick bite fever from the Centers for Disease Control and Prevention. Raw Living Radio Interview 3-show series in HD 2014 from the Earthshift Project. Common educational and informational research organization welcoming more participation.